Hi, my name is Lea Ormandy and I attend Manhattanville College in Purchase, New York. In this video, I will be presenting my senior thesis titled Monitoring of Oxygen, pH, and Nutrient Flux under Elevated Phosphate Conditions in Cassiopea xamachana. I'll start with a bit of background on the species. Cassiopea xamachana are commonly known as upside-down jellyfish and native to shallow coastal waters and mangrove habitats of the Florida Keys and Caribbean Sea. These jellyfish maintain a symbiotic relationship with photosynthetic zooxanthellae of the family Symbiodinium. The jellyfish take up nutrients in exchange for carbon photosynthates provided by the zooxanthellae. Pictured at the bottom of the poster are a few of the animals used for the lab experiments. Cassiopea can have a significant influence on benthic oxygen and nutrient dynamics in their ecosystems. The day and night cycle drives a constant flux in surrounding nutrient concentrations. Upside-down jellyfish take in ammonium, phosphate, and nitrate during photosynthesis and release these nutrients in dark periods. Research on nutrient exchange in Cassiopea is pertinent to environmental concerns such as anthropogenic or human-influenced nutrient loading, which along with increasing global temperatures spurs problematic jellyfish blooms. The introduction of abnormally high concentrations of limiting nutrients such as nitrogen and phosphate can cause a massive boom in jellyfish populations, the decomposition of which causes ecologically devastating hypoxic conditions. Understanding how Cassiopea react to high levels of limiting nutrients could help predict and control these blooms and allow us to monitor ecosystems with greater depth. Previous research has suggested Cassiopea to be useful as bioindicators, organisms used to indicate ecosystem health. The first goal of the study was to observe the effects of Cassiopea presence on oxygen, pH, and nutrient levels, and develop an apparatus for continuous monitoring of these parameters. The second was to investigate the effects of elevated phosphate exposure on Cassiopea using the continuous monitoring setup. The hypothesis for the first trial, regarding the effects of jellyfish presence, was that jellyfish will cause water-dissolved oxygen to elevate during light periods and decline in dark periods. Photosynthetic activity will deplete nitrate and phosphate concentrations, while pH will not change. The hypothesis for trial 2, involving the effects of elevated phosphate conditions, was that jellyfish incubated with added phosphate will deplete phosphate at lower quantities than those without addition, producing a net phosphate increase in the water. Nitrate levels will not be affected by phosphate addition. This diagram depicts the experimental design. Feel free to pause the video to read details on the materials and methods. One pH probe and one dissolved oxygen probe, both connected to a Vernier LabQuest tablet, was placed in each container. Dissolved oxygen and pH were continuously monitored on the tablets and recorded over a 48-hour period at a 4 sample per hour rate. The results of Trial 1 revealed that continuous monitoring proved advantageous over point sampling in monitoring water quality, and its capacity to reveal real-time trends in water parameters. Hypothesized, dissolved oxygen concentrations fluctuated in accordance with light and dark periods. The jellyfish, predicted to deplete nutrients, turned out to be nitrate and phosphate sources. Exposure to elevated phosphate in Cassiopea alters surrounding dissolved oxygen, pH, and PO4 levels. The results support the hypothesis that elevated phosphate lowers subsequent phosphate uptake. Trials could be conducted to monitor Cassiopea over more extended time periods. More frequent nitrate and phosphate samples could be taken to greater reflect nutrient flux. Further trials could be conducted to monitor Cassiopea over extended time periods. More frequent nitrate and phosphate samples could be taken to greater reflect nutrient flux data. Once again, the data collected in this research project contributes to the understanding of jellyfish nutrient dynamics that could be applied to issues such as pollution and jellyfish blooms. Thank you very much for listening.